Hello, this is chapter 2 where we talk about cells and cell division. We've previously looked at DNA replication where one chromosome goes to 2, or in the case of humans 46 chromosomes go to 92, and this happens during the process of cell division. So we're going to look at this in this larger context. This is a generalized eukaryotic cell, multiple components in here. If you are not sure about each of these components and their function, you should review your freshman biology notes. Diseases and traits are generally reflections of what is happening in a cell. So if we can understand this cell and what's going on, we can probably understand a disease. Of course, this being a genetics course, we're interested in specifically the nucleus. This is a transmission EM of a nucleus here. This is the nucleus. This is the nuclear membrane just around the outside. This is the cytoplasm outside. This area here is the nucleolus. The nucleolus is a subcompartment of the nucleus where there's a lot of ribosomal RNA transcription takes place. So this is the nucleolus, nucleus, and this is the nuclear membrane. If we zoom in to the nuclear membrane, it looks like this. This is a scanning EM of a nuclear membrane, and each of these is a pore. Each of these is a little hole in the nuclear membrane through which in and out go messenger RNA and proteins and anything else that needs to go in and out of the membrane. Over here on the right, this is again a transmission EM. This is the nucleus here, and on the outside of the nucleus we can see this stuff around here, and this is rough endoplasmic reticulum. And rough ER is called rough because it has lots and lots and lots of ribosomes. All these little dots here are ribosomes. So when a messenger RNA comes out of the nucleus, it can attach to one of these ribosomes and translate the protein immediately and put that into the endoplasmic reticulum for further processing. So this looks at a karyotype of a human cell. Humans have 46 chromosomes. We call that number a diploid number. 2n equals 46. The gametes contain 23 chromosomes, just half. The gametes, of course, are the eggs and the sperm. And the human chromosomes, human sex chromosomes here are X and Y. We can see those down here. Here's X and Y chromosomes down here. And the autosomes are essentially all the others. So all this lot is the autosomes, and these two are the sex chromosomes. So when you're replicating a cell, DNA replication occurs in a cell cycle. And this illustrates a cell cycle. In other words, we start off with one cell here, it goes around the cycle, and you end up with two cells. The time this takes varies depending on the cell. For a human liver cell, you could average about 24 hours. Mitosis itself, the actual aspect of mitosis, is just this green area around here. That is when the cell is dividing the DNA between the two new cells here. Everything else, something else goes on here. This is called interphase. Interphase consists of G1, S, and G2, and they take up a good portion, like 75 to 90% of the time. So let's look at each of these interphase stages in detail. The G1, which is over here, G1, this is where the cell doubles in size. If you think you're going to replicate a cell and divide it in two, then you have to double its size to start with, otherwise you're going to end up with two half-size cells. Similarly, you need to replicate all the organelles in that cell, otherwise you end up with only half the number in the next cell, so everything has to double. The S phase is where the DNA replicates. If you want to remember how when DNA replicates, it's the S phase synthesizes DNA. So each chromosome becomes two sister chromatids. It looks like this. Each chromosome, if you imagine a chromosome being depicted this way, after replication, we can now draw them like this. Still one centromere, therefore we call it still one chromosome, but each of these lines is now a sister chromatid. So we have two sister chromatids now. And G2 is preparing for division which occurs at the end of mitosis. Here's a cartoon of the chromosome, and this would be in G1 phase. This would be at the end of S phase, and you can still see the centromere attaches the two sister chromatids. Because there's one centromere, we still call it one chromosome. As soon as these sister chromatids split, which happens in anaphase, then these become two separate chromosomes. So we're going to look at the process of mitosis, and here is the summary. We start with interphase up here and runs all the way through to cytokinesis, where we double and the cell actually splits in two. And you can see there are a number of stages, and the primary stages are prophase, metaphase, anaphase, telophase. So what are these other things? What's pro-metaphase? Well, you have to remember that this is not a staged process. This is a dynamic process. One stage flows into the other. And so pro-metaphase is really just halfway between prophase and metaphase. So we'll look at the first stage of prophase. And one of the things here is that chromosomes condense and become visible. In interphase, the chromosomes are not visible to light microscopy. Then they condense down using nucleosomes and histones and become thicker and shorter and visible. The other thing that happens is the nuclear membrane needs to break down. That's this stage here. This is the nuclear membrane, right there. This makes sense because if you want to split the chromosomes to different parts of the cell, you have to get rid of the boundary, which is the nuclear membrane. And then finally, these spindle fibers here, spindle fibers, these begin to form, and these are microtubules of protein that will eventually guide the chromosomes around the cell. The next stage is metaphase. Metaphase, the chromosomes move and become arranged across the middle of the cell. We call this the equator or the metaphase plate. And this is mitosis, which means all these chromosomes line up in a row. And you can see one sister chromatid faces one side and the other sister chromatid faces the other side. There are spindle fibers attached from the centromere to the centrioles. In these cases, these are centrioles. On each case, and they will draw one of the sister chromatids to each side of the cell. So in anaphase, which is this middle diagram here, this is anaphase, spindle fibers are broken down by proteins called kinetochores, and these sister chromatids are split. You can see how they're together here. They're split, and one goes to each side of the cell. Now these sister chromatids are now individual chromosomes. So what is a kinetochore? What is this thing here? This is a metaphase chromosome. Here's the centromere, is it right in the middle? And these blobs either side are the kinetochore proteins. This is the kinetochore. What the kinetochore does is it degrades spindle fibers. So these lines here are spindle fibers. If you imagine the kinetochore as sort of like a Pac-Man, and it eats the spindle fibers, it's going to draw that sister chromatid in that direction. Same thing with this side. If we start degrading spindle fibers on this side, it's going to draw the sister chromatid in that direction. You're going to split it down the middle and have two chromosomes. Finally, in mitosis, we have a telophase stage, which is this one right here. And telophase here is the opposite of prophase. So the nuclear membrane reforms, chromosomes decondense, and spindle fibers by this time have disappeared as well. We still have essentially one cell with two nuclei now. So we need to divide this cell into two, and that's where the process of cytokinesis comes in. And this is cytokinesis here, where we split the cell into two. And as you can see here, this cell is beginning to pinch in the middle here and start to divide in two. So we have two daughter cells over here. These are called daughter cells. You can do cytokinesis in two ways, depending on whether you are an animal or a plant cell. If you're an animal cell, it's over here, then you do not have a cell wall. So what you can do is squeeze in one part of your cell and start constricting the middle of your cell. It's called a cleavage furrow. nucleus here. That's a cleavage furrow right in there. And it's essentially a line of actin proteins that wrap around the cell and start to constrict, and basically like pinching a balloon into two. And in a plant cell, you can't do that because they have a cell wall. Here's the cell wall going around a plant cell. And what you can do is make a new cell wall. And that's called a cell plate. A cell plate is made, which is a new cell wall which divides the cell into two. So human cells do not grow forever. They can divide about 50 times, which allows enough growth to get to an adulthood and to repair yourself and everything. But stem cells, of course, can grow forever, as do cancer cells. So if your cells do not grow enough, then you can get premature aging. And this famous photograph is of a gentleman called John Tackett. He died at the age of 16. He's here photographed at age 15. And this was back in 2003 with this photograph. And so the disease John Tackett was suffering from was something called progeria, and there's another disease called Werner syndrome. And this is where the cells do not keep growing and you prematurely age. So there are experiments that have been done, especially with sea urchin eggs, that have elucidated the function of cyclin proteins in the control of cell division. Here we have a cell cycle, G1, S, G2, mitosis across the top with prophase, metaphase, anaphase, telophase, and cytokinesis in it. And there are two checkpoints, one down here between G1 and S, and another one up here between G2 and M. And these two checkpoints check for certain properties of the cell. And what is it that these two checkpoints are actually checking for? So that was mitosis, where one cell replicates and becomes two identical cells. That's two genetically identical cells. There's another process of cell division called meiosis, and that is where a cell will replicate, but it will replicate such that you end up with 4 cells instead of 2, and these 4 cells can be genetically different to each other. And so meiosis produces 4 haploid cells, which, if you remember, in human chromosomes we have 46 chromosomes is our diploid number, which means over here at the end of meiosis there's going to be 23 chromosomes in each of these 4 cells. We accomplish this process by 2 mechanisms called meiosis 1 and meiosis 2. And each mechanism contains prophase, metaphase, anaphase, and telophase. There's metaphase, anaphase, and telophase, and we call them prophase, metaphase, anaphase, telophase 1, and prophase, metaphase, anaphase, telophase 2. So the point of meiosis is to produce a cell with half the number of chromosomes in it, called a haploid cell. And just to illustrate this, here are chromosomes. Here we have 6 chromosomes. So in this case, 2n would equal 6. 2n equals 6. And there are 2 of every kind of chromosome, 2 orange, 2 purple, 2 yellow. The haploid cell, so n equals 3, contains 1 of each of those colors. So 1 of these purples, 1 of those pinks, 1 of the purples, and 1 of the yellows. Which one you choose is random, and that's part of the process of meiosis. So where did your chromosomes come from? If this is you down here with your diploid number, where did your chromosomes come from? Well, one of them must have come from your dad, and one of them must have come from your mum. So there can be mums and dads, mums and dads, mums and dads, and so on. But which one do you pass on? Again, it's a random choice as to which one of these 2 parental chromosomes that you have you pass on. So you may pass on the parental chromosome on the left, the maternal chromosome in the middle, and maybe another paternal chromosome on the right.